Okay, good morning and welcome everyone to Psychiatry Grand Rounds. I'm Scott Wilkie, and it's my great honor to introduce Dr. Conor Liston from Cornell University, whose talk today will focus on probing and rescuing dysfunctional brain circuits in depression. There are perhaps few individuals better suited than Conor to give such a talk. Conor is an extraordinarily accomplished physician scientist and psychiatrist. He completed his MD, Ph.D. and psychiatry residency training at Rockefeller and Cornell universities. He then went on to pursue his postdoctoral research, working with two of the early pioneers in developing approaches to study synapses and circuits in vivo, Dr. Wenbao Gan at NYU and Dr. Carl Deiseroff at Stanford. Thereafter, he returned to Cornell to open his own laboratory, where over the last decade, he has overseen an incredibly impressive research program in circuits neuroscience. Throughout this time, he has remained an active clinical psychiatrist, and I think this is likely part of what enables Conor to do such unique and translationally meaningful work. You can see that a driving force behind Conor's efforts is a commitment to developing new approaches to improve treatment of psychiatric disorders. This has particularly been the case in terms of emerging therapeutic strategies for depression. And so I think the work that Conor will be telling us about today is both innovative and important for the future of depression treatment in psychiatry. So with that, please join me in welcoming Dr. Conor Liston, and please feel free to put questions that you have in the Q&A section for after the talk.
All right. Thank you, Scott, for that very generous introduction. Let's sec here. All right, you're viewing the not presenter mode, right, Scott?
Sorry, what's the question?
You can see my screen.
I can see it. Yeah, I think you're okay.
Right? Good. All right. Thank you. Thanks. And thank you for that very generous introduction. I'm really excited to be here. I'm sorry I couldn't be there in person. I have many friends and collaborators at UCLA, including Scott and his lab, and I hope I can come and visit in person sometime soon. So the work I'm going to present today, before I jump into that, actually, I'm going to just acknowledge some conflicts. I've served as a scientific advisor for Compass Pathways, Delix Therapeutics, Brainify, and Janssen Neuroscience, but I'm not going to be discussing any work connected with my role as an advisor to these organizations today. So the work I'm going to present is organized around understanding the mechanisms that control mood state transitions in patients with depression. Depression is this, by definition, fundamentally episodic psychiatric syndrome. Clinicians in the room know this. It's defined by these periods of low mood interposed between periods of wellness. And most work to date has focused on comparing a group of depressed individuals with a group of never depressed healthy controls, or doing something kind of analogous in a preclinical rodent model. And we've learned a lot from that work. But in order to get at the mechanisms that control transitions over time, we think a kind of complementary longitudinal approach is going to be important. And one really beautiful example of that comes from Bob Post and his colleagues, published almost 20 years ago. You see like an excerpt from one of their figures here. They just tracked and quantified mood in a group of patients on an individual basis, repeatedly over many weeks and months, up to a couple of years, I believe. And the results, three examples you see plotted here. Periods of depressed mood are below the x-axis, periods of hypomania or mania are above the x-axis, and 0 represents normal euthymic mood. And you can see in these three examples, there's lots to appreciate, but one of the obvious messages is that they just look different from one another. The dynamics that characterize this person's depression look quite different from the dynamics characterizing this second individual. This one, for example, has kind of characteristic lengths to their depressive episodes. This person's depressive episodes are shorter. They tend to switch back and forth more frequently. There are relatively fewer periods of prolonged euthymic mood compared to this individual, for example. And Post and colleagues identified still other examples of this. And so much of the work in my lab has been trying to understand what controls these transitions. Why does a person get depressed today as opposed to last week or next month? When they get depressed, what controls how long they stay depressed? And conversely, when they get better, are there perhaps one set of mechanisms that are important in initiating their recovery? And are there others perhaps that are important in sustaining their recovery over time? And to do that, we use repeated fMRI assessments in human patients and repeated two-photon imaging in mouse models to try to understand the mechanisms that control those transitions. Undoubtedly, many mechanisms are involved, but converging evidence from a variety of studies suggests that one of them might involve the remodeling of these structures called postsynaptic dendritic spines. For people unfamiliar with spines, you can think of these as structural markers of synapses. Most spines contain synapses. Not all spines contain synapses. I'll be using the terms kind of interchangeably today, but I'm happy to talk about the distinction if that's of interest in the Q&A. We've known for a while, mainly from studies in fixed tissue, that chronic stress, which recapitulates behaviors reminiscent of some aspects of depression in rodents, that chronic stress causes a loss of these spines and synapses in the prefrontal cortex. And that antidepressants like ketamine tend to do the opposite, increase in synapse density in the prefrontal cortex and other areas. And so it's been tempting to speculate that these two things are causally related, that the formation of new synapses might be required for inducing that switch from stressed to not, or from kind of depressed to healthy. And yet it's also possible that that they're not causally related, that synapse formation and changes in behavior are merely correlated with some third factor that's actually causally important. And getting at that question is more than just an academic one. It also has potential clinical translational impact. If you think that forming new synapses is causally driving antidepressant effects, then that stands to reason that other interventions that form new synapses might be useful as antidepressants. And so the work I'm going to tell you to start was driven by Rachel Modosava, an amazing grad student who's now at Harvard, Mitch Murdoch, now at MIT, and Pooja Parekh, a postdoc in the lab, who is actually on the job market and doing amazing work. Please check out Pooja's work. They set out to ask these three questions. First, does ketamine, a rapidly acting antidepressant, does it selectively rescue stress-related synapse loss in the prefrontal cortex? Second, how does synapse formation influence circuit function and behavior? And third, does the formation of new synapses play a causal role in mediating these effects? And not to bury the lead, I'll tell you that we found that it did play a causal role, but not in the way we expected. And I'm going to unpack that for you over the next 5 or 10 minutes. Before I jump into the results, I just want to orient you to the methods that we're using. So we are imaging the remodeling of postsynaptic dendritic spines in the living brain through repeated two-photon laser scanning microscopy, imaged through a chronically implanted microprism into the contralateral hemisphere of the brain. This is to scale. These prisms are about the size of Abraham Lincoln's nose on a penny, very small, but in a mouse brain, they're substantial. But this prep offers some advantages. We're implanting it in the contralateral hemisphere and imaging across the midline, and that means that the hemisphere we're imaging from is relatively intact. A second advantage is that we can get a lot of light in and out of the tissue, and that means we can generate these relatively high-resolution images that allow us to resolve individual dendritic spines and synapses. And using vascular landmarks and contours of the prism, we can return again and again to the exact same place with micrometer precision, like you see here. And we can ask what impact stress and antidepressants like ketamine are having on the turnover of spines and synapses in these Phi1 yellow fluorescent protein expressing transgenic mice. So let me show you the first set of experiments. Here we implant a prism, we give the mice some time to recover, then we image the mice before and after various chronic stress manipulations. The data I'm showing you here come from chronic exposure to corticosterone, the mouse version of cortisol in the drinking water. It's associated with a marked increase in spine elimination. That's not surprising based on what I told you we already knew from the fixed tissue studies. But because we're doing this in vivo, we can begin to ask more targeted questions, like whether there's evidence that spines that are lost tend to cluster together on individual dendritic branch segments. And that's relevant because we know that one of the functions of dendritic branch segmentation is to segregate functionally homogeneous synaptic inputs, meaning that all of or most of the projections from region A might tend to synapse on branch 1 but not 2, whereas most of the projections from region B might tend to synapse on branch 2 but not branch 1. And what we found was that there was this strong bimodal distribution. Most dendritic branch segments showed spine loss rates comparable to controls, but some showed markedly elevated spine loss rates, sometimes up to 10-fold the normal number of controls, suggesting that there was some logic to which spines were being lost. We also found that lost spines tended to be spatially clustered. I'm not showing that data, you can take my word for it. And we're currently working to figure out what those spines might be connected to, but we think this data suggests to us that it's not random, that perhaps particular projections are being targeted for pruning, and perhaps when I return, I'll have an answer for you as to what those projections are. Next, we turned to ask, what does ketamine, an antidepressant, what does ketamine do? These are the same mice imaged before and after chronic stress, then treated with ketamine or a vehicle injection, and then imaged again 24 hours later when we know ketamine has antidepressant effects in humans. And we see a similar pattern, but this time for spine formation, a big increase in spine formation that's bimodally distributed and spatially clustered. And that led us to ask whether it might be the case that ketamine is acting in a targeted way to restore spines that were lost after stress. And to test that, we classified each spine that formed after chronic cort exposure as a restored spine if, like this blue one, it formed in a position where a spine used to exist but then disappeared, or a de novo formed spine was a spine that formed in a position where we didn't observe a spine in either of the previous two images. What we found is that ketamine had a big effect on spine restoration, restoring many of the spines that were lost after stress to the same or nearly the same position that they existed in the baseline condition. This occurred much more than you'd expect by chance based on the observed rate of spine formation. Now, what I've shown you is kind of the glass half full view of these data. The glass half empty view is also here if you look a little harder. This shows you that about half of the spines that form after ketamine treatment are restored spines, which means that about half of them are not restored spines. And that in turn means that although the number of spines that has formed after ketamine has kind of undone the effects of stress. In other words, the number of spines, the density of spines after ketamine treatment looks like it would in unstressed mice. And yet it is not the case that the full configuration of spines that was present before the stressor is fully reversed. Some spines are restored, some are not, and we think that might be relevant for perhaps for the recurrence of depression-related behavior later, and I'm going to return to that in a minute. Shifting gears a little bit, I want to tell you just a bit about what we've learned about the functional correlates of these changes. Here, we're doing the same experiment, imaging mice before and after chronic stress, treating them with ketamine, imaging them again a day later. But instead of imaging spines, we're imaging circuit function using a genetically encoded calcium sensor, GCAM6, which causes these neurons expressing the sensor to fluoresce when the cell becomes active. And you get these kind of Van Gogh Starry Night-like images that give you a real-time readout of how active each neuron is at a given time point. And using algorithms developed by others, we can segment these images, including some others here at UCLA. We can segment these images into individual neurons and extract calcium signal time series, which are kind of readouts of activity. And we can ask lots of questions about what stress and ketamine are doing to those activity patterns. One of the most basic questions you might ask, and the first one we asked, if we know that stress is causing a loss of synapses and ketamine is restoring some of those synapses, you might predict the structure of correlated activity in this field of view would change. In other words, that functional connectivity, as measured by correlations between these neurons, would change after stress and after ketamine treatment. And that's indeed what we observed, and that's illustrated here in two different ways from one representative mouse. At the bottom, you see correlation matrices where each element depicts functional coupling between a pair of cells, and warm colors denote pairs of cells that are strongly functionally coupled. And you can see without any fancy statistics that there's just a loss of warm colors after chronic court treatments compared to the baseline imaging session. And that's depicted in another way in these raster plots. Here, each row is a cell, left to right is time, and each element is colored based on how active the cell is at a given time. And one of the things that just kind of stood out to us unexpectedly, we didn't really design the experiments to detect this, but we saw that a lot of the activity that occurs at baseline occurs in the context of these multi-cell ensemble events, where many or even most cells become active at about the same time. And after chronic court treatment, the occurrence of these ensemble events is reduced in frequency, and when they occur, they tend to involve fewer cells. We found that ketamine rescued both of those effects. This is one representative mouse. We see this very consistently in all the mice we image. I'm not going to show you that data. In fact, in the interest of time, I'm going to skip over a few slides here. Ask me to return to them in the Q&A if you're interested. I just want to make sure we have plenty of time for questions. So let's turn now to talk about the behavioral correlates of the functional and synaptic changes that I just described. We've explored this in a variety of ways. I don't have time to tell you about all of them today. We've looked at the role of these ensembles in conditioned fear learning, in cognitive flexibility, in social hierarchy perception. I'd refer you in particular to Tim Spellman's amazing work. This was published in Cell a couple of years ago. Tim has now started his own lab at the University of Connecticut. He's doing awesome work. Check that out. Don't have time to tell you about it today. Instead, I'm going to just tell you briefly about work from Rob Fecho, looking at the role of these ensembles in effortful reward-seeking behavior, and from Rachel Motasava, looking at motivated escape behavior, and trying to get at that central question of cause and effect. I think in the interest of time, I'm just going to refer you to Rob's paper. It was published in Neuron at the end of last year. Check it out if you're interested. But summarizing a lot of work, Rob, who's now a postdoc at Princeton, Rob adapted a task that was developed in rats for use in mice. I think Scott and his lab are also doing great work in this area. This task presents mice with a choice to navigate left or right in a T-maze to get a large reward by climbing over a barrier, or a small reward without any barrier. And we can adjust the ratio of the rewards and the size of the barrier to see how much incentive is required to motivate a mouse to choose the big reward coupled with effort. And what Rob found, summarizing a lot of work, is that neurons in the anterior cingulate cortex that project to the nucleus accumbens encode a signal that anticipates rewards and is modulated by effort requirements. And by optogenetically silencing these cells, he showed that this signal is required for reinforcing future decisions to expend effort to obtain large rewards. The signal is also sensitive to stress, greatly diminished after chronic court exposure, as is their kind of effortful reward-seeking behavior. And we're currently looking at how antidepressants influence that behavior. Before I switch to telling you a bit about our human work, I want to conclude by addressing kind of the key question I posed at the beginning, which is how changes in spines, changes in circuit function, and changes in behavior are related to one another in terms of cause and effect. And our approach here focused on a really simple behavior motivated by a beautiful study from Melissa Warden, showing that medial prefrontal neurons that project to the dorsal raphe are important for driving and kind of controlling response to an acute stressor. They drive motivated escape behavior in tests like the forced swim test and the tail suspension test. I think of these tests, you can interpret them in lots of different ways. I like to interpret them based on, what we're actually observing, which is that mice and rats tend to alternate between two kinds of behavior when confronted with a stress, like a forced swim challenge. They alternate between struggling to escape or remaining perfectly immobile. And the kind of post hoc just so story for why a mouse might remain immobile when confronted with a stressor is that many of their predators rely on vision to catch them. If you're a mouse, almost everything you see in the world is a potential predator. Even rats prey on mice. And so predators like birds of prey, like cats, might be less likely to see the mouse if it remains perfectly immobile. and perhaps that response is adaptive in some contexts. We know that stress tends to increase immobility and decrease motivated escape behavior, and antidepressants tend to do the opposite. And Melissa showed that optogenetically driving these cells increases struggling escape behavior. And so we just wanted to ask whether something similar might be happening here. Again, summarizing a lot of work in one kind of representative figure panel, we found that in accord with Melissa's work that these cells, their activity was related to struggling versus immobility behavior. Specifically, we see an increase, pink bands here in this photometry recording denote periods of struggling, white bands denote periods of immobility. And you can see there's an increase in activity during struggling, and it actually precedes and predicts shifts from immobility to struggling pretty reliably by about a second. And during chronic stress states, the occurrence of those ensemble events, like I showed you before, is reduced in frequency. And so on average, animals tend to switch back and forth less frequently, and they remain immobile for longer periods of time. And we think that this behavior, which in the past has been relevant for screening for at least for SSRI like antidepressants, and also for ketamine's antidepressant properties, might tell us something about some of the antidepressant behavioral effects. And so to get at cause and effect, we did two kind of simple experiments, one relatively simple, one a little more complicated. The first is just looking at the time course of events here. If you think A causes B, A should come before B. And if instead you find that A comes after B, that tells you something about cause and effect. And so in this first experiment, we treated mice with chronic corticosterone, imaged them before and after, and then in the hours to days up to a week after ketamine treatment, doing repeated assessments involving spine imaging, calcium imaging, or behavioral testing. And what we found was not what we expected, but the data were very clear. And you might see other examples of that from my presentation today. Science sometimes leads us in interesting and unexpected directions. We found that the effect of ketamine on behavior was very rapid, much more rapid than what we observed for spine formation. You could detect effects 3 hours after treatment. I would note that if we had gone into the literature a little more carefully, we would have found that others have seen this too. Most groups tend to begin looking at these effects a day after ketamine treatment, but others have looked over shorter time periods and have also seen this. We now know that these effects are detectable even faster than 3 hours, probably more like 30 minutes. And the effect on spines don't begin to become detectable until 12 hours after treatment, peaking a day after treatment. So that told us right there that the formation of new spines, at least in these neurons that we're imaging from, could not be required for initiating ketamine's antidepressant behavioral effect, at least not on this behavioral assay. And incidentally, we found that the effects on circuit function paralleled the effects on behavior and preceded the effects on spines. So you'd see this restoration of correlated activity and of ensemble events 3 hours after ketamine treatment, well before the 12-hour time point when we see spines forming. So that caused us to go back and kind of rethink our hypothesis. And one hint for how to understand this came from this scatter plot, which showed that the maintenance of restored spines 2 days or 1 week after treatment was fairly tightly coupled with the maintenance of ketamine's antidepressant behavioral effects on immobility in the tail suspension test. And so that led us to hypothesize that perhaps it's the case that new spines and synapses aren't required for initiating the behavioral effect, but they might be required for sustaining it over time. And to test that, we teamed up with a group at the University of Tokyo led by Haruo Kasai and Haruhiko Bito. They developed a synaptic optogenetic tool, which they call Activated Synapse Photoactivatable RAC1. The name is a bit of a mouthful, but the basic idea is very simple and elegant. This is a blue light sensitive probe whose expression is targeted to a sparse set of recently potentiated synapses and newly formed spines. And upon prolonged exposure to blue light, the probe changes conformation, initiating the RAC1 signaling cascade, and through interactions with the actin cytoskeleton, causes spines expressing the probe to shrink and to collapse. And so they can basically, in a selective way, optically delete newly formed spines and synapses. And using this approach, they showed that the formation of new spines in the forelimb area of motor cortex was not required for an initial learning event of a motor skill, but it was required for maintaining a memory of that motor skill over time. And so we wanted to ask whether something similar might be happening with ketamine. We treated mice with chronic cort, spines disappeared. We treated them with ketamine. Some of those spines came back. We photoactivated 12 hours after ketamine treatment. You'll recall this is a time point when we saw a lot of spine formation occurring. And then we asked what impact that had on spines and behavior 2 days later, also 1 week later. This is validation data showing you that this intervention was sufficient to disrupt the effects of ketamine on spine formation. And then this is the critical behavioral data. It shows you that as expected, chronic cort increases in mobility in this test. Ketamine reverses that effect. But if you photoactivate 12 hours after ketamine, the effect wears off. And we also had an important negative control experiment showing that if you photoactivate 3 hours after ketamine treatment, and you'll recall that 3 hours after treatment, we didn't observe any spine formation related to ketamine. So what we're doing here is deleting another set of spines unrelated to ketamine treatments. And you see this has no effect on behavior. So what this tells us, we think, is that the formation of new spines after ketamine treatment, there's something special about those spines in particular, and they are important for maintaining ketamine's antidepressant behavioral effects over time. And that in turn implies that other interventions aimed at boosting the survival of those spines or enhancing the restoration of lost spines might be useful for prolonging the antidepressant effects of ketamine. And that's something we're testing because as clinicians in the room know, ketamine is an amazing treatment, so powerful in many ways. But absent some other intervention, if you give people 1 dose of ketamine and nothing else, most of them will get depressed eventually, again, on average, within a couple of weeks. And so other interventions that might be paired with ketamine for prolonging its effects might be useful. Okay, I'm going to shift gears now and tell you a little bit about our work in humans, and then there'll be plenty of time for questions at the end. Again, I may skip through a couple of slides just to save time. So one of the big questions we've been interested in the lab for some time now is understanding a different form of heterogeneity. I described at the beginning how different patients exhibit very different dynamics in their mood state transitions over time. But we also know that different patients present with very different kinds of symptoms. And we diagnose depression today when a person presents with 5 or more of these 9 symptoms. And I think it stands to reason. There are at least 256 unique combinations of symptoms that meet criteria for a major depressive episode. That's not even accounting for the fact that some of these criteria contain opposites of themselves. And I think it's intuitive that someone who's suffering from intense anxiety, physical agitation, sleeping 4 hours a night, and lost 30 pounds, this person may not have the exact same biological problem as someone who's sleeping 20 hours a day, can't get out of bed, physically slowed, not anxious, just profoundly anhedonic, gained 30 pounds with intense carbohydrate craving. These people may not have the exact same biological problem, and yet they get the same treatment. And this is the part in the talk that I think will be obvious to clinicians again, but it bears repeating and some emphasis. What I'm about to tell you about our work to rethink how we diagnose depression shouldn't be taken as discouragement from seeking help. In a way, it's a miracle that our treatments work as well as they do for these very different people. And they do work well for lots of patients. They just don't work as well as we would like. And there's some consensus in the field that one of the reasons they may not work as well and one of the obstacles to developing more effective treatments may relate to how we diagnose depression and lumping very different people into these big categories. Of course, we're not the first to think about this problem. There's a very rich literature dating back decades, and painting with very broad strokes, the kind of dominant approach for many years in subtype discovery efforts was to group patients on the basis of their symptoms, looking for kind of symptoms that tend to co-occur in subsets of patients, and then asking whether those subtypes are associated with biomarkers that might be useful for diagnosing them or for selecting treatments. And we've learned a lot from that work. Some of these, like seasonal depression, is one that's seeped into the public lexicon that we here in New York, maybe more than you in LA, are aware of at this time of year. But the fact remains that it's been hard to translate, with some notable exceptions notwithstanding, it's been hard to translate these clinical subtypes into biomarkers that can be used for informing clinical decisions in widespread clinical practice. And so our approach has been to try to flip this upside down. Instead of grouping patients on the basis of symptoms, let's try to group them on the basis of biology, and then ask whether those putative biological subtypes are associated with different clinical symptom profiles or different treatment outcomes. The biological measures we focused on are these resting state fMRI measures of functional connectivity, which are actually not unlike the functional connectivity measures I described in our two-photon imaging earlier. They're based on the seminal discovery by Bharat Biswal and Mark Rakel many years ago now, that the brain at rest exhibits these spontaneous fluctuations in the bold signal, which you see in this video, and regions of the brain that are strongly connected to one another tend to fluctuate together. And you can measure how coupled they are by correlating those low frequency fluctuations in like long resting state fMRI scans. I'm going to be referring to connectivity in several different contexts in the remainder of the talk. When I talk about connectivity now, this is what I mean: functional connectivity, correlations in the bold signal. It's influenced by monosynaptic structural connectivity, but it is, of course, also influenced by other things. Those are not the same. The work I'm about to describe was made possible by Jonathan Downer, a colleague at the University of Toronto, and Faith Gunning and Mark Dubin, my colleagues here at Cornell. I want to acknowledge them up front, as well as a host of others who shared their data with me and made this work possible, sometimes before the data were even publicly available. I'm very grateful to them. The work wouldn't have happened without them. We knew we would need a lot of data to do this work. The first data set that we used comprised 711 subjects, a little less than half of whom were actively depressed with unipolar major depressive disorder. We used this data set to discover the subtypes that I'm about to describe, and to train machine learning algorithms for making various predictions about the subtypes. Machine learning aficionados know that these methods are kind of notorious for overfitting to idiosyncrasies in the data that they're trained on. So it's really important to have an independent replication data set, and that's what this second data set came in. Just again, to orient you to the kind of data we're using, we gathered up all this resting state fMRI data from many different groups. We used the same functional parcellation to extract bold signal time series from different regions of interest spanning the entire brain. And we compute a correlation between each region and every other region. You see one pair depicted here, and repeat that process for all regions. And you get a matrix that looks like this. Importantly, it's kind of a whole brain map of functional connectivity. This matrix contains 33,000, more than 33,000 unique functional connectivity features. Presumably, most of these have nothing to do with depression. And so we knew we needed a way to express these in a kind of low dimensional space, and to hone in on the features that were most important in depression. If we were to cluster on all of them, we might find clusters, but they'd probably have nothing to do with depression. The method we settled on is called canonical correlation analysis. It's sort of similar to PCA. It looks for linear combinations of connectivity features in the fMRI scan that explain a lot of variance in the connectivity, subject to the additional constraint that they should also explain and predict individual differences in particular sets of clinical symptoms. And what we found was evidence of at least 2 brain behavior dimensions explaining individual differences respectively in anhedonia and anxiety and insomnia. We now know that there are actually more than 2. We have strong evidence of at least 3. There are probably more than that. Follow up on that in a moment. We also know that there's overfitting in this data set. This was an issue identified early by us and by others, but this is an issue that can be solved by incorporating regularization into the canonical correlation analysis procedure. We and others have used this and similar techniques with different kinds of success. I'd refer you Rebecca Price, Logan Grosnick's work, Ted Satterthwaite's work, many others as well. This just shows you that by incorporating regularization, you can get good performance in held out data. These violin plots, the blue plots are training data, the green plots are test data. And you can see that the correlations in training data and test data are only a little different. So we know that this works well. We've now replicated this in other data sets. I'd refer you in particular to a former postdoc, Katie Dunlop, who's now an assistant professor at the University of Toronto. Her paper kind of replicating and extending this approach just came out in Biological Psychiatry. Please check out Katie's work. Amanda Booch, a former grad student, has had success with a similar approach in autism, which I don't have time to tell you about today, but please check out Amanda's work. Having identified these two brain behavior dimensions, we then clustered patients in this 2D space. And we found, using hierarchical clustering, strong evidence of at least 4 subgroups. Now, I hasten to add what should be obvious from this scatter plot, which is that this clustering isn't like the clustering that you see in single nucleus RNA sequencing data. It's not like these are super discrete groups. There are subjects who live near the boundaries between these groups. But there is more clustering here than you'd expect by chance. And we don't think that this is kind of the last word or best solution to the problem of subtyping in depression. Rather, what I want to try to persuade you of is that this particular solution is useful, that you can make valuable predictions from it, and that this general approach holds promise. And I'm going to show you a couple of examples of that to conclude. So this shows you the maps of what's abnormal in functional connectivity in each subtype. Warm colors denote connections that are strengthened in depression. Cool colors denote connections that are weakened in depression. And what you can see without worrying too much about the details of what regions are involved is that these 4 maps just look very different. The DSM assigns a single label to 4 groups of patients with very different functional connectivity profiles. They, in turn, are associated with different kinds of symptoms. For example, the 4th subtype is associated with high levels of anxiety, insomnia, and anhedonia. The 2nd subtype is associated more with kind of low energy and fatigue. We identified subtype differences in a host of other symptoms, ranging from anhedonia to psychomotor slowing, anxiety, different forms of insomnia, anergia. But you don't need an fMRI biomarker to diagnose depression or to characterize a person's symptoms. That wouldn't be especially clinically useful. But it would be very valuable to have a biomarker that might tell you something about treatment decisions. We set out to validate this approach with respect to predicting treatment outcomes in a way that was motivated by this figure. This shows you the neuroanatomical distribution of connectivity features that differ by subtype. And these nodes are scaled by how different the subtypes are with respect to the connectivity features in this area. And you can see that one of the nodes with the biggest differences is this area of the dorsomedial prefrontal cortex, which is not the most commonly used kind of FDA-cleared target for repetitive transcranial magnetic stimulation, but it is a well-validated target. We know it works for depression. And so we wanted to test whether it might be the case that these subtypes, which have very different connectivity at this target site, might respond differently to stimulating that target site. And we found that they did. Subtype 1 was very responsive to TMS. This is, for aficionados, high frequency, mostly 10 hertz, a little bit of intermittent theta burst stimulation of dorsomedial prefrontal cortex, times 6 weeks in a kind of conventional protocol. The first subtype was very responsive. The second and fourth subtypes were not at all responsive. And the third subtype showed this kind of intermediate level of responsiveness. These differences were sufficient to train machine learning algorithms, which you can think of as kind of neuroimaging biomarkers, statistical classifiers for predicting who would respond and who would not respond to this kind of treatment. We can do pretty well with connectivity features alone. We can do better if we add in the subtype diagnosis. And although these results based just on clinical symptoms are statistically significant, they are not that much better than chance. I'd also just emphasize here that the performance you're seeing in these confusion matrices are for leave one out cross-validation, which tends to overestimate performance, in a new sample. We don't think that you should use these numbers as a reliable gauge of like the actual quantitative performance that you're going to observe in a big community sample. Rather, what we think this is telling us is that this approach is useful for predicting dorsomedial prefrontal TMS response and worth developing further. But ideally, what you'd have is a biomarker that helps you not just predict response to one treatment, but choose between two treatments. And this approach is motivated by beautiful work from Helen Mayberg's lab showing that you can use subgenial cingulate connectivity measured by fMRI to predict differential response to either cognitive behavioral therapy or to an antidepressant medication for depression. Which suggests that in principle, this approach that Helen developed might be useful for choosing between these two treatments. And so we wanted to ask whether something similar could be developed for TMS, choosing between the dorsomedial prefrontal target, which I just described, and the more standard left dorsolateral prefrontal target. And we now have biomarkers based on the work that I just described that allow us to formulate predictions on an individual basis about response to these two targets. And we're currently testing how well they perform. This is a clinical trial led by myself, Jonathan Downer, Faith Gunning, and Ben Zebley, the director of interventional psychiatry program at Cornell, as well as Nolan Williams, a close collaborator at Stanford. We think this work is really important, probably for obvious reasons, like you don't know how well a biomarker is going to perform if that biomarker was trained in retrospective data until you kind of nail it down and test it prospectively. But perhaps less obvious is that even a biomarker that is like 100% accurate could still not be that clinically useful. And that kind of conundrum is illustrated in these three kind of hypothetical scenarios. In the far right example, where your response to one treatment is highly correlated with your response to another treatment, it doesn't matter whether your biomarker is 100% accurate. Everyone is either going to respond well to both treatments or not respond well to either treatments. And so you can just roll the dice and give them when it doesn't really matter. On the other hand, if reality were more like this kind of left scenario here, where everyone is a strong responder to one treatment, but not the other, you could in principle get everyone better if you just knew which treatment to give them in the first place. And based on our modeling, we think reality is kind of somewhere in between. And these people in the lower left and upper left and lower right quadrants are the ones we're really going to be able to help. We're currently testing this in a randomized controlled trial. We do have some funds to fly people to New York. If you know people in the New York area or even outside the New York area who might be good candidates for this, please reach out. We are actively recruiting. And in the last maybe 5 minutes, I'm going to tell you about some really exciting work that circles back to the original question I posed at the beginning about an effort to understand mechanisms that control mood state transitions through longitudinal assessments. This is work led by Chuck Lynch and Emmanuel Elbaud, two former postdocs, now assistant professors at Cornell, where we do repeated imaging in patients as they cycle in and out of depression. This is one example of this. And I have to commend Chuck, especially, and also Emmanuel for leading this work. It's really kind of a extremely laborious and heroic effort. They have now scanned and repeatedly assessed a small group of people 40, 60, up to 80 times over the course of several years, allowing them to kind of map very carefully how functional connectivity and symptoms change, to correlate those time series, and to ask whether changes in connectivity might even predict future changes in clinical symptoms. And I can show you a couple of examples to close. The general approach was motivated by new findings from a host of researchers at WashU, at Harvard, UCLA, Stanford, showing that the brain at rest is organized into these functional networks but there are actually individual differences in the precise topography of how these networks are organized, which I think of with analogy to human faces. We all have two eyes and a nose and a mouth, and if you average these faces together, you get something that looks readily recognizable as a face, but this average face glosses over all the individual differences that are so important in how we use faces. And we now know that brains are similar. That everyone, for example, has a yellow network, which is, in this case, the frontoparietal control network, in about the same place in the lateral prefrontal cortex, but the precise boundaries of that network actually vary quite substantially. And you can imagine how, stimulating that area defined anatomically might lead to very different responses in a TMS study, and how these individual differences in network organization might be associated with differences in cognitive functions, in depressive symptoms, in risk for depression, et cetera. And so the first thing Chuck and Emmanuel wanted to do was just ask, for the first time to my knowledge, whether there are differences in the topography of these networks in patients with depression. And they were kind of uniquely suited to do so with this densely sampled data. We have tens of thousands of minutes of scanning in these patients. And one thing really stood out to us, frankly, kind of unexpectedly. This is work from Chuck and Emmanuel up on bioRxiv. You can check it out. They found that one network and only one network was really dramatically expanded in the brains of patients with depression. And that was this frontostriatal salience network, which occupies the anterior cingulate cortex, a bit of the lateral prefrontal cortex, and the anterior insula mainly, also aspects of the striatum. And you can see, again, without any fancy statistics, this is just bigger than this by a lot. This is one patient, one representative example who's healthy and one representative example who's depressed. This is kind of a group representation of those findings. These are big effects in our hands. Individual functional connectivity features in depression tend to have effect sizes, abnormalities in depression on the order of 0.15 to 0.25. These are like an order of magnitude bigger. We replicated this effect in multiple data sets. The effect was sufficiently large to train classifiers to diagnose depression fairly reliably, just solely based on network topography. We identified, interestingly, three modes of expansion involving encroachment on differing networks. I don't have time to tell you about it. Ask me if you're interested. And that led us to ask whether it might be the case that this network is kind of growing and shrinking as a person gets depressed and not depressed. And our data set was very well suited to answering that question. And again, it gave us a very clear answer, which refuted our original prediction, which is that the network really does not change. It's really rock solid stable in patients with depression. You can see that in this one individual person. This is study visit 1, 5, 10, and 13. They're depressed in 1 and 5. They're not depressed in 10 and 13. You can see the boundaries are really remarkably similar. This is kind of more examples from 6 different patients. The percent surface area of the cortex that the salience network occupies really doesn't change across study visits. It's completely unrelated to how depressed a person is at a given time point. These are 3 individuals assessed 13, 62, or 39 times. It doesn't change with treatment. It doesn't vary with symptoms across individuals. So, in the previous slide, I showed you correlations with symptoms within an individual. This is across about 150 some individuals, no relationship. And it also is unrelated to the number of prior episodes that a person has had. So this is all kind of like not what we expected. And again, led us to kind of rethink our approach here. Perhaps it's the case that this salience network expansion isn't driving mood symptoms in like a real-time kind of state-dependent way, but it might instead be a marker of risk for becoming depressed. And to test that, we turned to the ABCD sample among 10,000 kids who were scanned at ages 10 and 12. We identified 60 who were never depressed at ages 10 and 12 and went on to become depressed at age 13 and had sufficient data to do precision functional mapping. And our prediction was that the salience network might be expanded in these kids before they even got depressed consistent with the idea that it might be a marker of risk for depression. And that's indeed what we observed, a notable expansion of the salience network in these kids who went on to become depressed, not shown here. We don't see that in kids who don't go on to become depressed. So we think, it emerges early in life and it may confer risk for depression. We need to explore that further. And the last thing I'll show you is that even though salience network expansion, is stable and doesn't drive mood state switches acutely, it is in fact a case that changes in functional connectivity between nodes within the network are important in predicting current mood states. And to assess that, we turned to these subjects who we had densely sampled. And we just asked whether changes in functional connectivity between nodes of the salience network were associated with changes first in anhedonia, which is what you see plotted here. And we found that they were. The correlation is a moderate one, 0.37, but it's very reliable. This is within one individual. This is across individuals. Effect size is remarkably similar. And it's specifically related to connectivity between the anterior cingulate and the nucleus accumbens nodes of the salience network. And you'll recall that I told you at the beginning that in our mice, this anterior cingulate nucleus accumbens circuit appears to be important for supporting effortful reward-seeking behavior. We found that this was unrelated to anxiety, that another circuit was related to changes in anxiety. And importantly, we found that you could not only predict current anhedonia symptoms based on functional connectivity in that circuit, but you could actually also predict the occurrence of future anhedonia symptoms before they emerged. So this is a cross correlation depicting connectivity at time 0 with anhedonia at time 0. And you see this correlation around like minus 0.4. And you can see that we can also detect that with a lag of -1, meaning correlating connectivity today with anhedonia next time you come in. And so we think, this is one step towards establishing a potential causal relationship between changes in connectivity in this circuit and changes in the specific depressive symptom domain. I want to stop at that. Before I take questions, I just want to highlight all the people who contributed to this, especially the folks in blue who did all this work, and my collaborators depicted here, especially the people in red who contributed directly to some of the data I presented. And I'll stop with that. Questions?
Thanks, Connor. That was awesome. Very interesting, exciting work. If people have questions, they should put them in the Q&A feature of the Zoom, and I will convey them to Connor. I guess I'll just start with one of my own, Connor, which is, you showed that there's this loss of, I'm thinking more about the ketamine stuff, this loss of dendritic spines when mice are exposed to stress, and then, some of them are restored, and then there are some new ones that are forming. Have you looked at, or do we know what's happening with the presynaptic partner of that spine? Is the synapse actually lost, or is it there in some sort of, less stable form?
Yep, I love that question. We've thought a lot about that too. We don't know for sure, but our hunch is very aligned with, I think, what you're speculating about. I think a related question, is like, how do these spines know how to come back in the exact same position, so to speak? And we think it may have to do with the presynaptic partner of the spine. We're only imaging, as you know, the postsynaptic side here of the spines, not the presynaptic boutons. We actually have done some presynaptic bouton imaging. That's a work in progress. Happy to kind of share that offline if people are interested. But we do have reason to suspect that the presynaptic side is also changing, but that some remnants may persist and may regulate and facilitate the formation of new spines in particular positions. We know from Ryohei Yasuda's work and others that spines also release diffusible factors. So neighboring spines might influence the propensity of a spine to form in a particular position. That might have something to do with it as well. But you're absolutely right. We think that there may be some remnant of the synapse, either on the presynaptic or postsynaptic side, or potentially both, that is influencing that. But I will say that we do know, that synapses are being lost, and functional synapses are being lost, and not just spines. And we know that from Ph.D. 95 staining from some patch clamp electrophysiology work. But you're exactly right. I think there's probably something left over that is guiding spine restoration.
Yeah, that makes sense. So we have another question from an audience member, which is kind of related to this, which is, if we think that the spines aren't causing the like kind of initial antidepressant like effect, the question is whether you have speculations about what might be the mechanism of that initial, response.
We do. We have a whole other paper that we're preparing to submit. We hope it will be up on bioRxiv maybe later this month. Following up on that, summarizing a lot of work. Again, many mechanisms are surely involved, but we think a key mechanism in initiating the effect involves either direct or indirect activation of mu opioid receptor signaling in somatostatin interneurons. We know from work from teams at Alan Schatzberg, Nolan Williams, Carolyn Rodriguez at Stanford, I believe there may be replications of this coming out soon, that if you co-treat with ketamine and naltrexone, that mu opioid receptor antagonist, that this blocks the antidepressant behavioral effects of ketamine. And we think that is related to mu signaling, specifically GIO coupled GPCR signaling in SST cells, which hyperpolarizes those SST cells, disinhibiting projection neurons, restoring ensemble events, and that this is really critical for initiating the antidepressant effect. And we have now quite a lot of data implicating that mechanism in particular. Happy to talk offline in more detail about that if people are interested.
Great. And then we have another question, which is, I'm going to sort of try to interpret this. The question is, does immobility in mice correlate with anhedonia and struggle with anxiety? And so, I think perhaps the question is, related to what do these behaviors in mice actually mean? You talked a little bit about that, but do you think that this can be related to psychiatric symptoms in some way?
Yeah, that's a really good question, and I know like you, Scott, and probably others on this call also think about these questions. I don't have like the one right answer. I'll tell you what like my kind of speculation is. I mean, I hesitate to say that immobility is a readout of anhedonia and struggling is a readout of anxiety. But I think the questioner's point is well taken. That like, for example, we know from other work from Melissa Warden and Avi Avikari here at UCLA and many others, that that a mouse's response to an acute stressor, that struggling response, is strongly motivated by context, possibly by internal states like anxiety, and that like what the mouse does, like in an elevated plus maze, for example, a really anxious mouse will tend to huddle in the corner of the enclosed arm of the maze. But if you get it really, really anxious, it will actually walk out on the open arm and jump off in an attempt to escape. So it can be tricky, is my point, to interpret these behaviors. And I hesitate to go as far as to say that immobility is correlated with anhedonia and struggling with anxiety. But there might be, some modest relationship between the two. And I think there's cool work out there that's being done to kind of unpack that further. I think that's the best answer I can give at the moment about that.
Yeah, that's a hard question.
Yeah.
I think maybe we have time for one more really quick one. I guess I'm just curious, like you've looked at these kind of forced swim tests, tail suspension tests, another question is like about what, could you look at different behaviors? So for example, are the dendritic spines required for some effect of antidepressants that you're just kind of not capturing with that assay? Have you looked at any other behaviors?
Yep. Love that idea too. We're beginning to do that. In Rachel's paper, we showed sort of like something related, but a little different, which was that like if you delete those spines, like I showed you, that interferes with the maintenance of its effects on immobility, behavior, and tail suspension. And we also showed that it actually has no effect on sucrose preference, which I think actually also sort of gets to the previous questioner's question. They are distinct things. I think the best way to do that is really like more with behaviors of the sort that you're using in your lab, Scott, and others too, that are more structured and more interpretable. Like if you have a question about behavior per se, then assays that are designed more as screening tools might not be the right approach. We think they all have value, but in short, I would say my guess would be yes, that like that spines in different regions are going to be required for different kinds of behaviors, and the story is going to be complicated. And we're trying to do that, for example, with that effortful reward seeking task, with this cognitive flexibility task, with other tasks. That's a great idea too.
Great. Awesome. Well, thank you so much, Connor. I think we are wrapping up now, but appreciate you coming on Zoom. Hopefully we'll see you in person soon.
Fantastic. Thanks for having me. Yeah, hope to see you guys soon.